In your NCERT textbooks, I know you're finding certain complicated terms, isn't it? Like compatible, incompatible, and pollen-pistil interactions. Now, let us very quickly simplify these terms and understand what's it all about. Now, coming to the compatibility, we are talking about the pollen grains that are compatible and incompatible here. And how is it going to be simplified? That is very easy. What you're seeing here, now let us assume we have a pistil over here. So, let's assume this is the plant nerium and here we have the pistil, right? Now here you have the stigma onto which this pollen falling on, isn't it? Now what is a compatible pollen? Now let's assume this one where you are seeing there's a pollen grain upon falling onto the stigma successfully forming a pollen tube, right? The pollen tube is entering into the embryo sac and also the fusion of the sperm cells and the egg cells also happen, right? Sperm cell and the egg cell. Now, what is happening here? A pollen grain which is successfully able to form a pollen tube and eventually leading to fertilization to happen is called as a compatible pollen grain. Now, what is that incompatible means? That which is not forming all this, isn't it? Now, let's assume there's another pollen grain. Now, yes, this is a nerium plant, isn't it? Now, very obviously, this is a nerium pollen which is able to form a pollen tube. So, it's compatible, right? Now, let's assume there is a neem pollen grain which is falling onto the stigma of this nerium plant, right? Now, here what's happening? There is no formation of the pollen tube. So, that is incompatible very clearly. In some cases, pollen tube is forming, but it is not growing further, isn't it? Now, also in some cases, it might grow, but it is unable to enter the embryo sac. If it enters also, there is no fusion of male and female nuclei happening there, right? So, such pollen is incompatible pollen, right? Now, we understood about compatibility and incompatibility, and let me tell you, in some plant species, like in case of nerium even the plant's own pollen can be sometimes incompatible. Now, let us understand who is going to decide all this incompatible or compatibility between the pollen and the stigma. Now, what is happening here? You're seeing a pollen grain is falling onto the stigma, isn't it? What happens immediately? The stigma is going to release certain chemicals. Right, on receiving these chemicals, pollen is also going to secrete some more chemicals. So there is a exchange of chemicals happening here, like how you and I talk, right? So there's a dialogue happening between the stigma and the pollen. So this is nothing but the pollen-pistil interactions. Now this chemical dialogue is going to happen from the point of time when the pollen grain has fallen onto the stigma till the fertilization happens. And throughout all these, there's a chemical dialogue or exchange of chemicals going on, and that is called as pistil pollen interactions, right? I hope that's clear for you. And also, let me tell you, we have also made a clear simplified video about pollen development and also the embryo sac development. The link is in the description. Go through that before you understand this episode. All right, now let's understand about something very interesting. How is a tricellular pollen and bicellular pollen behaving in case of the pollen tube growth? Right, now we already studied about this, that there is like more than 60% of the angiosperm which are going to release the pollen in bicellular pollen state, and less than 40% would do that in tricellular pollen state. Right, now let's understand how both of these pollen grains are behaving in terms of the pollen tube growth. Now coming to the first one, let's understand here. So we have a tricellular pollen grain over here, isn't it? Now here you're seeing that I already have told you, isn't it, that there would be certain chemicals that are released from the stigma. Here also you are seeing the chemicals are released by the stigma, upon receiving which the vegetative cell, you're seeing the vegetative cell is going to respond by forming a pollen tube. Now here you are seeing the vegetative cell nucleus, right? You're seeing the vegetative cell nucleus, and here you have got the sperm cells. Now what's happening here? The vegetative cell is forming the pollen tube and the sperm cells are present, and successfully you are seeing the pollen tube is entering into the embryo sac, and there is a fusion of male and female nuclei happening here, right? So that's happening in the tricellular pollen. Now, what about the bicellular pollen state here? So, here we have a bicellular pollen grain. Now, definitely this is also compatible. So, upon receiving the liquid or the chemical from the stigma, here also you are seeing the vegetative cell is going to form the pollen tube. Right. Now, what you're finding here, you're finding the vegetative cell and also the generative cell. And now only one small difference happens here. Like we have seen in previously in the tricellular pollen state, isn't it? There were two sperm cells. Now, what happens here? The generative cell. So, here we have enlarged here. The generative cell is going to undergo a cell division forming what? Forming two sperm cells. Right. Now, what is happening here? You're seeing after this division, you're seeing the pollen tube is growing into the embryo sac and the sperm cells are going to go for the fertilization. Now, one hard and fast rule that you have to remember is that pollen tube will definitely contain two sperm cells before it enters the embryo sac and before the fertilization happens. So, that's about the difference of pollen tube growth in tricellular and bicellular pollen states. Now let's go ahead and understand about the double fertilization and triple fusion. Now we have seen in tricellular and also in bicellular pollen states, we had two sperm cells. Right. Now, what is the fate of this? How is it going to go ahead? Let's understand. So here, you are seeing that tube the pollen tube is entering through the micropyle, isn't it? Now here we have the sperm cells. Through the micropyle, it enters the synergid. Right. Now we have the synergid over here. Now, here, why only the synergids? Why not through any other point? The pollen tube and the synergid interaction is also very important. Here, in the synergids, we have certain apparatus, certain specialized apparatus, which are called as filiform apparatus. Now, how does it know? How does the pollen tube knows that there is the embryo sac present over here? How does it knows? How does it know the location? It's only because of these filiform apparatus which are going to secrete a specialized chemical which is going to direct the pollen tube towards the embryo sac, and that's how the entry happens. Now, once the pollen tube enters the synergid, what happens? This synergid would die, right? And the tip of the pollen tube would rupture, releasing the sperm cells. Now, after that, the sperm cells would move to the space between the egg cell and the central cell. Right, so we all know that this is the central cell over here, isn't it? So between the egg cell and the central cell, we have the sperm cells. Now, what's the next event? Very clearly, one of these sperm nuclei fuses with the egg cell and the other sperm nucleus will fuse with the polar nuclei. Right, now very clearly, two events of fertilization are happening here. One's happening here, isn't it, where the sperm nucleus is fusing with the egg cell nucleus, and here the other sperm nucleus is fusing with the two polar nuclei. So that is why we call this as double fertilization. Right, double, two events of fertilization or two events of fusions happening. So we call this as double fertilization. And something for you to remember as well is about syngamy, which is nothing but the fusion of egg cell and the sperm cell, which you are seeing here, the event of syngamy which is happening. Right, now what's the fate of this ones now? Now here you are seeing this is the zygote nucleus, isn't it? Previously, this was the egg cell, isn't it? Now, we call this as the zygote cell and this is the zygote nucleus. Now, what's happening to this? What is this event leading to? It is leading to the primary endosperm nucleus, primary endospermic nucleus. And now this central cell which you are seeing here would be called as primary endosperm cell. Right. Now also something very important for you to remember is triple fusion. Where did you just see the triple fusion? Here, isn't it? You are seeing 3 nuclei participating in the fusion. One from the sperms, isn't it? This is one sperm nucleus and 2 polar nuclei, isn't it? 1 and 2. So 1, 2 and 3. So that's why we have the name triple fusion, which is fusion of 2 polar nuclei with the sperm cell. Right? So that's about double fertilization, triple fusion, formation of zygote nucleus, primary endospermic nucleus and the primary endosperm cell. Now, let's understand what is the Ploidy change after fertilization. This is very important in terms of your entrance, right? This is something very confusing and also very easy for you to remember. What is before? What is the ploidy before fertilization? Let's look at that first. So here you are seeing the egg cell is haploid. The polar nuclei are also haploid. Each of the sperm cell is haploid, right? So this is before fertilization. Even here you can see the same thing happening. There's no fertilization so far. Now after fertilization, you are seeing the haploid sperm nucleus and the haploid egg cell nucleus, isn't it? They are fusing to form the zygote nucleus, isn't it? So this will be diploid, right? Here you are seeing the other sperm nucleus fusing with 2 polar nuclei which are haploid, isn't it? So 1, 1 and 1. 1 plus 1 plus 1, that is 3, which is 3, and this is triploid primary endosperm nucleus formation over here. That's it. So before the fertilization and after the fertilization, if you remember it with that difference, it is very easy for you to get reminded of these, each of the ploidy, whenever it is asked in your entrances. Now, let's understand. So, how this ploidy changes after fertilization. So, let's very quickly remember that in the tabular form. So, what's happening here? In case of the egg cell, we call this egg cell, isn't it, before fertilization? And after that, the nucleus was called zygote nucleus and the cell is called as zygote. It's no more the egg cell. And this zygote further develops into embryo, which is also diploid in future. Now, central cell. Now, in the central cell, after the fertilization, what's happening? We will call that as primary endosperm cell, which is going to be formed into the endosperm, which is triploid. Now, coming to some PYQs, let's very quickly revise about whatever we have seen so far through these PYQs. So, coming to the first question. So we have in angiosperms the haploid, diploid and triploid structure. So haploid, diploid, triploid. So they are asking this in a sequence of a fertilized embryo sac. So how are those? Antipodal, synergids and primary endosperm nucleus. So that is definitely not happening. This is 1 and this is 1. So this is not diploid. Synergids, zygote, primary endospermic nucleus. Haploid, diploid, triploid. So this would be our right answer. Now moving to our next question. What is the fate of the male gametes discharged in the synergid? We understood that male gametes are discharged into the synergid when the pollen tube tip is ruptured, isn't it? So what happens to that? Let us understand that one fuses with the egg and other fuses with the central cell nuclei. One fuses with the egg, others degenerate. So definitely not happening. All fuse with the egg, no. One fuses with the egg and others fuses with synergids, no. So definitely our right answer would be A. One fuses with the egg and other one with the central cell nucleus. Now, double fertilization is very simply, very easily, we can answer this one. Fusion of 2 male gametes with 1 egg. So we have 2 events of fertilization, isn't it? So definitely it is not explaining about the second event. Fusion of 1 male gamete with 2 polar nuclei. It's also telling about only one event. Fusion of 2 male gametes of pollen tube with 2 different eggs. Not the scenario. Now syngamy. What is syngamy? We understood, isn't it? Fusion of male and female nuclei and also triple fusion, isn't it? So there is syngamy and triple fusion happening. So D would be the right answer. Now the next one, double fertilization is exhibited by gymnosperms, algae, fungi and angiosperms. Clear answer, angiosperms. D would be the right one. So coming to this one, let's understand through which cell of the embryo sac does the pollen tube enter the embryo sac, right? Not through the egg cell, definitely not through the central cell. Now we have 2 synergids named over here? Yes, definitely it is synergid. The answer would be like, what is it now? Is it persistent or is it degenerated? So we know we have two synergids, isn't it, with the filiform apparatus. Now let's assume there's a pollen tube that is entering to this synergid, right? Now what is happening to this synergid after the pollen tube enters? It's going to die, isn't it? This is going to degenerate, and this other synergid is going to be persistent. Now very clearly we can understand through which synergid is the pollen tube entering. It's not the persistent synergid, it's the degenerated synergid, right? So C would be the right answer. Coming to the one, a diploid female plant cell. We have a diploid female plant cell, a tetraploid male plant are crossed. Now what are they asking? The ploidy of the endosperm, right? Now from the tetraploid male plant, half of the ploidy will be the sperm nucleus, isn't it? So we have 2n of this one. And diploid female plant, we already know there were two polar nuclei, isn't it? It was 2n. Now when we have the fusion here, we have 2n plus 2n, that is tetraploid. Endosperm is the ploidy of the plant when the cross is happening between diploid female and a tetraploid male plant. In angiosperms, pollen tubes liberate their male gametes into, very simple, we have the pollen tubes. Where are they releasing the gametes? Into the synergid, definitely, isn't it? Not the egg cell, there's a fusion happening here. Antipodals are not involved. Central cell also there would be a fusion. So we have the synergids to be the right answer. Next, endosperm is formed during the double fertilization by, very quickly, two polar nuclei and one male gamete, one polar nuclei, one male gamete, ovum and the male gamete, two polar nuclei and two male gametes. They're asking about the endosperm, isn't it? So very easily, two polar nuclei, isn't it, with one male gamete. So this was the triple fusion which we discussed about, isn't it? So A would be the right answer. Now, the role of double fertilization in angiosperms is to produce what? Because of the double fertilization, we are having what? There is a triple fusion happening there, because of which we get the endosperm. Now, if an angiospermic male plant is diploid, so they're saying male is diploid this time and the female plant is tetraploid. Right? Now, what is the ploidy level of the endosperm? So, you here, because of the diploid male plant, you are getting haploid sperm cell, isn't it? Because of the tetraploid female plant, we are going to have a central cell would be also tetraploid. There would be 2n and 2n of polar nuclei. Now, when the polar nuclei are fused together, we are going to call that as central cell, right? Now, here, when these fusion is happening, N, 2N and 2N, that would be the pentaploid of the endosperm, right? Now, moving ahead, double fertilization is fusion of, very simple. Double fertilization is fusion of, and it's a direct question over here. We have two eggs, definitely not. Two eggs and polar nuclei, one male gamete with egg, other with synergid, definitely not. One male gamete with egg and the other with secondary nucleus. So we have already discussed about the secondary nucleus in the central cell, isn't it? So these are the polar nuclei which are together. We call that as the secondary nucleus. And Syngamy, very simple, fusion of the gametes always. Don't get confused with any other term. Always the fusion of gametes only represents Syngamy, not the fusion of cytoplasms or anything. It's the fusion of gametes only. Now, moving ahead, double fertilization leading to initiation of endosperm in angiosperms require what? Right? What is that? It is fusion of one polar nuclei with second male gamete only. No. Fusion of two polar nuclei and the second male gamete. there are no 4 or more polar nuclei. All of the above is also not right, isn't it? So the fusion of two polar nuclei and the second male gamete would be the right answer. Now coming to the last one. So fertilization involving carrying of male gametes by the pollen tube is called as siphonogamy. How will you remember this? Tube is always, remember, tube means siphon. So they're asking about pollen tube. So we will call that as siphonogamy, right? So that's it for this episode. If you vibed with us, don't forget to subscribe to this and also share this content with your friends. Give us a like if you understood everything very clearly. And don't forget to follow us on Instagram. And here I am signing off, and I will be back with another episode to simplify another complicated concept. Thank you.